Hi, my name is Teddy Corcoran, and in this video I'll be discussing the downstream effects of receptor activation, specifically with regards to short-term versus long-term outcomes. Receptor activation via ligand binding can have a myriad of downstream effects on cells. While the exact effects of this activation may vary by the type of ligand, receptor, cell, and other factors, they can generally be classified into two main categories: short-term and long-term. In the short term, these activation events can trigger cellular signaling cascades that alter cell properties in response to the signal. Often, this type of cascade is initiated via a phosphorylation event, though other signaling molecules such as calcium may be used instead. These effects are usually localized, though some can be more global, and are only suited to endure for a short period of time. On the other hand, some aspects of receptor activation result in much longer-term responses for the cells involved. In this case, a signaling cascade is typically involved in the activation of various transcription factors. These factors, localized in the nucleus, have enormous potential to alter the long-term traits, behaviors, and even viability of cells in which they are activated. In this video, we will explore several cases of receptor activation in the nervous system during development, making note of the short and long-term effects, as well as some of the differences and similarities between the two. One example that illustrates this concept well is nerve growth factor, or NGF. NGF is vital to early neuron survivability. Its existence was first hypothesized as a result of experiments that demonstrated larger target regions increased neuron survival, with the opposite being true for smaller target regions. Similarly, embryos carrying sarcomas showed hyperplastic growth of sympathetic ganglia. Importantly, these and other experiments showed that apoptosis appeared to be the default fate for neurons that were unable to interact with their target tissues. They indicated something about the target region was inducing additional neuron growth and survivability, specifically an active secreted factor later determined to be NGF. This factor was shown to induce rapid tyrosine phosphorylation when plated with PC12 cells, and was later determined to be the ligand for an orphan receptor tyrosine kinase, or RTK. Getting into the short-term effects of the receptor, which you may have guessed from the name, RTK activation results in rapid phosphorylation of tyrosine molecules. Phosphorylation events are transient, though, and cannot explain the increased longevity and growth of neurons previously observed. Here, we get into the long-term effects of NGF receptor activation. Apoptosis proceeds through a signaling cascade, meaning numerous steps must be activated or inhibited in sequence in order for cell death to occur. Additionally, some of these pathways may be pro-apoptotic, promoting cell death, while others are anti-apoptotic, preventing apoptosis. Here, tyrosine phosphorylation functions as the first step in an anti-apoptotic pathway. Downstream, this phosphorylation triggers additional protein activation, culminating in the activation of transcription factors such as CREB. These transcription factors regulate gene expression, meaning they have a much longer-term effect on cells. Activation of these transcription factors as a long-term outcome of receptor activation serves to inhibit apoptosis, allowing a trophic factor to influence cell survivability on a genetic level. Another example of the short and long-term effects of receptor activation can be found in synapse development and maintenance. Early in development, there are an abundance of synapses, far more than are functionally necessary in an adult human. As a result, pruning or elimination of some of these synapses is critical to normal development. Though many factors are involved in this process, activity is particularly important in deciding which synapses will be maintained and which will be removed. In excitatory neurons, two types of receptors are particularly important in the mechanism of use it or lose it: AMPA receptors and NMDA receptors. Both are glutamate receptors present on the postsynaptic neuron. While AMPA receptors fire in response to glutamate only, NMDA receptors are also voltage dependent, with a magnesium ion blocking the receptor channel under normal conditions. If the neuron has been previously depolarized, say by the prior activation of AMPA receptors, this ion will be freed. If a subsequent release of glutamate then interacts with NMDA receptors, they will open in addition to the AMPA receptors and allow an influx of calcium into the cell. This influx of calcium and the events that immediately follow can be classified as the short-term effects of this receptor. Calcium release triggers activation of calmodulin-related pathways, phosphorylating local proteins, including AMPA receptors. Phosphorylation of these receptors results in their stabilization and facilitates their incorporation into the membrane. Short-term, the receptor activation helps to increase the presence of AMPA receptors at the postsynaptic site, However, as we mentioned previously, phosphorylation is a transient event and would not lead to the long-term potentiation needed to promote synaptic longevity. This is where we get into the long-term effects of NMDA receptor activation. The release of calcium is not only concentrated locally. This receptor activation also triggers more global release of calcium from the endoplasmic reticulum. In this way, calcium acts as a secondary messenger. Calcium influx triggers a signaling cascade in a variety of different pathways, including RAS, which is involved in the cell survival pathway, adenylate cyclase and CAMP, as well as additional calmodulin pathway. All of these pathways are involved in activating transcription factors, but the CAMP pathway is of particular interest in synaptic plasticity. Here, CAMP activates pKa, which then phosphorylates a molecule called CREB, located in the nucleus. This molecule binds to a Cre site upstream of target genes, causing the recruitment of other factors, culminating in polymerase being recruited to the promoter region of a gene, initiating gene transcription. These transcription targets can have a variety of effects on the original synapse. They may lead to the formation of additional synapses near the original, alter the characteristics of presynaptic vesicles packed with signaling neurotransmitters, or increase the number or alter the properties of postsynaptic receptors.
On this note, I thought it may be interesting to examine how deficits in a specific gene involved in this process can dramatically change the effects of receptor activation, both short and long term. The gene is UBE3A, which encodes a protein involved in AMPA receptor degradation.
Experience-driven neuronal activity induces UBE3A transcription, and UBE3A then regulates excitatory synapse development by controlling the degradation of ARC, a synaptic protein that promotes the internalization of AMPA receptors. Deficits in this gene lead to an increase in ARC expression, thus decreasing AMPA receptors on the postsynaptic surface. As we just discussed, AMPA receptors are vital to long-term potentiation and synaptic strengthening both directly and indirectly via their role in activating NMDA receptors. In the long term, clinically, deletions or other complications in UBE3A results in two major disorders, Prader-Willi syndrome and Angelman syndrome. Both involve significant cognitive issues, including language impediments, motor skill impairment, microcephaly, and general mental retardation. In short, significant differences from normal development.
UBE3A is an excellent example of the importance of receptor activation in neuronal development. Deficits in this gene lead directly to deficits in normal synapse receptor properties, causing significant developmental issues.
Overall, the two examples we covered today, early neuronal survivability and synaptic plasticity, highlight the short and long-term outcomes of receptor activation. In the short term, for both examples, we see transient activity immediately following this activation with tyrosine phosphorylation for NGF and calcium release for NMDA receptors in the synapse. Both are a rapid response to the ligand binding, but neither has the potential to alter cell properties permanently. For this, we turn to the long-term outcomes of receptor activation. The short-term effects of these receptors serve as the first step in a signaling cascade that ultimately activates transcription factors. These transcription factors can in turn activate other genes with the potential to fundamentally alter cell properties, all as a long-term result of the initial ligand binding and receptor activation.